Hello everyone, welcome to Biotech Notebook. In this video, we are going to see last 5-year GATE Biotechnology Microbiology questions. The first question is, match the type of bacteria in group 1 with the respective growth properties in group 2. The first option is halophile. So, halophiles grows at high level of salt. So, generally it is found in salt lakes, saline soils and salted food products. Next option is piezo-philes. So, piezo-philes grows optimally at hydrostatic pressure, high hydrostatic pressure. Okay, so hydrostatic pressure means pressure above the atmospheric pressure, that is pressure is greater than 1 atmosphere. Piezo-philes found deep down the ocean. The next one is mesophiles. Mesophiles grows optimally between 20 degree and 45 degree centigrade. That is, these mesophiles grow at neither too hot nor too cold. Okay, it grows at a moderate temperature between 20 degree centigrade and 45 degree centigrade. The next one is xerophiles. Xerophiles grows best at low water activity. Okay, so it grows at low water activity and generally it is found in cracks and pores of rocks. So xylophiles grows at a high level of salt, that is 3, and piezophils optimally at a high hydrostatic pressure, that is 4, and mesophiles grows optimally between 20 degree and 45 degree centigrade, and xerophiles grows best at low water activity. Okay, so 3, 4, 1, 2. Okay, so 3, 4, 1, 2. So option A is correct. Next question, match the virus in group 1 with the type of genome it contains in group 2. Okay, you can classify viruses based on the presence of nucleic acid in it. So you can classify viruses into two broad types. One is DNA viruses and RNA viruses. And this DNA viruses further classified into double-stranded. DS means double-stranded DNA viruses and SS, that is single-stranded DNA viruses. Okay, same RNA viruses also further classified into double-stranded RNA viruses and single-stranded RNA viruses. So in the question, group 1 has some examples for DNA and RNA viruses and group 2 has type of genome it contains. The first option, T4 bacteriophage. So T4 bacteriophage is example for double-stranded DNA. So second option, Q, SARS-CoV-2, that is severe acute respiratory syndrome coronavirus 2. So which has single-stranded RNA as a genome. Okay, then Pseudomonas phage 5-6 has double-stranded RNA, and 5X174 bacteriophage has single-stranded DNA. Okay, so T4 bacteriophage has double-stranded DNA, SARS-CoV-2 has single-stranded RNA, then Pseudomonas phage 5-6 has double-stranded RNA, 5X174 bacteriophage has single-stranded DNA. So 3, 4, 1, 2. Okay, so 3, 4, 1, 2. So option A is correct. Next question is the correct sequence of events during bacteriophage infection of a bacterial cell is. Okay, so here the correct answer is option A. So the first step in bacteriophage infection of bacterial cell is landing. Okay, so in this step, bacteriophage lands on the surface of your bacterial cell. Okay, so this is your outer membrane of bacterial cell and inner membrane of bacterial cells. Okay, so first step is landing of your bacteriophage on the bacterial cell, and the second step is attachment. Okay, in attachment step, bacteriophage attaches to the bacterial cell wall at a specific receptor site, okay, by the tail fibers. So this takes place by the tip of the virus tail. Okay, the tip of the virus tail becomes attached to the cell surface, okay, via a specific receptor site on the cell surface. Okay, this attachment is very specific that certain viruses and susceptible bacteria have complementary molecular configurations at their opposing receptor sites. So this process of attachment is also called as absorption. So next step is tail contraction. Okay, so tail contraction leads to penetration of your bacteriophage. So tail contraction means the tail sheath present in the tail contracts. This is your tail sheath. See the length of the tail sheath in your second stage and in the third stage. So this tail sheath contracts during infection to less than half of its initial length. So the contraction of this sheath derives the tail tube through the outer membrane and creating a channel for the viral genome delivery. The next step is penetration and unplugging So your contraction of tail sheath derives the tail tube through the outer membrane and also inner membrane, and it creates a channel for the viral genome delivery. So here the phage head and remaining component remains outside the bacteria. One tail of bacteriophage enters into the cell wall and cell membrane. Then it ejects the DNA into the bacteria. So this is the last step, DNA ejection. So option A is correct. First step is landing, then attachment, then tail contraction. Tail contraction leads to penetration and unplugging, then DNA ejection. So the next question, in binomial nomenclature, the name of bacterial strain is written with the first letter of dash word is being capitalized. Let's see what is binomial nomenclature and rule of binomial nomenclature. Okay, so binomial nomenclature is a biological system of naming the living organism, in which the name of the organism has two parts. The first part of your name is termed as genus and second part is termed as species. Okay, then the rule of binomial nomenclature is the first letter of your first word, that is genus, always capitalized. Okay, first letter of first word should be always capitalized, and first letter of second term, that is your species, should not be capitalized and it is always written in lowercase. Then the scientific name is always italicized, or you should underline the word. So in the question they are asking, a bacterial strain is written with the first letter of dash word is being capitalized. First letter of first word is being capitalized. So the answer is option A first. Next question is, the type of nucleic acid present in lambda-phage is. Lambda-phage is a bacterial virus, also known as bacteriophage, which infects bacterial species E. coli. And this lambda-phage has head, tail and tail fiber, and this head contains the phage's nucleic acid, double-stranded circular DNA. So the option A is correct. The type of nucleic acid present in lambda-phage is double-stranded DNA. Next question is, the specific growth rate of a yeast having a doubling time of 0.693 hour is dash in R power minus 1. Okay, so in the question they have given doubling time of yeast, that is TD is equal to 0.693 hour. They are asking to calculate specific growth rate of a yeast in R power minus 1. So you have to calculate mu. Mu is nothing but specific growth rate. TD is doubling time. Formula to calculate doubling time is TD is equal to 0.693 divided by mu. You can rewrite this equation as mu is equal to 0.693 divided by TD. So in the given data they have given TD as 0.693 hour. So mu is equal to 0.693 divided by 0.693 hour. So 0.693 will cancel it out. So you will get mu is equal to 1 hour power minus 1. Next question is, a bacterial strain is grown in nutrient medium at 37 degrees centigrade under aerobic conditions. Okay, that is in the presence of oxygen. The medium is inoculated with 10 power 2 cells from a seed culture. If the number of cells in the culture is 10 power 5 after 10 hours of growth, the doubling time of the strain is in hour. So here they have given the time duration of your growth is 10 hours. That is, you are growing your organism for 10 hours. So T is equal to 10 hours. You are inoculating with 10 power 2 cells from a seed culture. That is capital B. Okay, so capital B is number of bacteria. Okay, number of bacteria at the beginning of a time interval. And they have given number of bacteria at the end of the time interval. That is, after 10 hours, your number of bacteria is 10 power 5. That is small b. So small b is equal to 10 power 5 cells. Okay, small b is number of bacteria at the end of the time interval. Capital B is number of bacteria before the time interval, here 10 hours. Okay, and they are asking the doubling time of the strain is. So doubling time TD, or you may write as generation time G. So the formula to calculate generation time or doubling time TD is equal to T divided by N. Okay, so here T is time interval in hours, so it is given 10 hours, and N is number of generation time. Okay, number of generation time, that is defined as number of times the cell population doubles during the time interval. Okay, so during this time interval, how many number of times the cell population doubles is known as number of generation time. Okay, so in the given data they have not given n. Okay, so we have to find out n. Okay, so this n can be calculated by using this equation, that is 3.3 log small b divided by capital B. Okay, small b is given data, capital B is given data. So by substituting small b and capital D, you can find out n. So as I already said, capital B is equal to number of bacteria at the beginning of a time interval and small b is number of bacteria at the end of the time interval. So substitute this formula in this equation. So you will get G is equal to T divided by N. So N is 3.3 into log of small b divided by capital B. So just substitute the given data. So T is 10 hours, 3.3 into log, small b is 10 power 5 cells and capital B is 10 power 2 cells. So you will get 10 divided by 3.3 into log 5 minus 2, you will get 3. So 10 power 3. So 10 divided by 3.3 into log 10 power 3. So 10 divided by 3.3 log 10 power 3 is log 1000 is equal to 3. Then 10 divided by 3.3 into 3 is equal to 9.9. So 10 divided by 9.9 is equal to 1.0101. So here the unit is in hour. So question, which one of the following is correct about microbial growth medium? Okay, so the first option is Luria bertani broth. So Luria bertani broth is a synthetic medium. Okay, so this is wrong because Luria bertani broth is a complex medium. So let's know the difference between synthetic medium and complex medium. Okay, synthetic medium is a medium in which all chemical components are known. Okay, well defined medium. Whereas complex medium contains extracts and digest of yeast, meat or plants. Okay, you can't define the medium composition. Okay, the chemical composition of the medium is not known. This Luria bertani broth contains tryptone, yeast extract and NaCl. Okay, so it contains yeast extract. Composition of yeast extract is not defined, so LB broth comes under complex medium. The next one is nutrient broth is a defined medium. Nutrient broth also a complex medium because it contains yeast extract, peptone and NaCl. Okay, so it also contains yeast extract. It is also an example for complex medium. Okay, defined medium is nothing but a medium in which all the chemical components are known. This option is also wrong. So next option is Sabert dextrose agar is a differential medium. Okay, this option is also wrong because SDA, that is Sabert dextrose agar, is a selective solid media. It contains dextrose, peptone and agar. Let's know the difference between selective media and differential media. Selective media allows certain type of organisms to grow and it inhibits the growth of the other organisms. Okay, this Sabert dextrose agar allows the growth of pathogenic and non-pathogenic fungi. Okay, mostly molds and yeast, and it inhibits the growth of many bacteria. Whereas this differential medium is used to differentiate closely related organism by using specific biochemical properties of that organism. Generally, this medium contains some dye or some chemical component which is used to differentiate a closely related organism. The fourth option, trypticase soy agar, is a complex medium. Yes, this option is correct. TSA is a complex medium which contains pancreatic digest of casein, soybean digest, glucose, NaCl and dipotassium phosphate. We can define the composition of pancreatic digest of casein and soybean digester. So this medium is complex medium. So the correct option is option 4, trypticase soy agar is a complex medium. Decimal reduction time of a bacterial strain is 20 minutes. Then what is specific death rate constant in minute power minus 1 is? Okay, first let's see what is decimal reduction time or D value. D value is the amount of time that it takes at a certain temperature to kill 90% of the organism being studied. Okay, after an organism is reduced by 1 D, only 10% of the original organism remain. Okay, so specific death rate constant you can be calculated by using this formula, that is ln of xt divided by x naught is equal to minus kd into t. X naught is initial population and xt is number of population at time t, and kd is specific death rate and t is time. So here negative sign indicates there will be a decrease in the number of microbial population. So in the problem they have given decimal reduction time t is equal to 20 minutes, and here if your initial cell concentration is 100 cells per ml, then after your decimal reduction time, that is here t is equal to 20 minutes, you will have only 10 cells per ml. Why? Because here you are reducing viable bacteria by 90% at a defined temperature. Okay, so 100 minus 90 will give 10 cells per ml. So ln of xt divided by x naught. So xt is 10 divided by x naught is 100 equal to minus kd into time is your decimal reduction time is 20 minutes. Okay, so kd is equal to ln of 10 divided by 100. 1 by 10 is equal to 0.1. So minus ln of 0.1 divided by 20. Ln of 0.1 is minus 2.3023 divided by 20. So this minus will cancel it out. So 2.3026 divided by 20 is equal to 0.1151. kd is equal to 0.1151 minute power minus 1. Next question is, which of the following antimicrobial agents is or growth factor analogs? Okay, first you should know what is growth factor. Okay, so growth factors are organic compounds such as amino acids, purines, pyrimids and vitamins a cell should have for its growth. Okay, a growth factor analog means these are chemicals which are structurally similar to bacterial growth factor, okay, but which do not fulfill its metabolic function in the cell. Okay, so a cell use growth factor for its growth, but growth factor analog prevent the growth of the cell. The structure of growth factor analogs are similar to growth factor, thereby it reduces the growth or it blocks the growth of the bacteria. So growth factor analogs can be used as antimicrobial agents. Okay, so antimicrobial agents means it will prevent the growth of the organism. So here the answer is 5-fluorouracil, isoniazid and sulfanilamide. Okay, these three are growth factor analogs which are structurally similar to pyrimidine derivatives. Okay, whereas tetracycline is antibiotic which inhibits the protein synthesis in bacteria. A 0.1 ml a liquid of bacteriophage stock. Okay, so your volume of phage stock is 0.1 ml. Having a concentration of 4 into 10 power 9 phages per ml. So concentration of your virus particle, that is here bacteriophage, is 4 into 10 power 9 per ml. 4 into 10 power 9 phages is added to 0.5 ml of E. coli culture. So your target cell is here E. coli culture. So the volume of E. coli culture is 0.5 ml. Having a concentration of 2 into 10 power 8 cells per ml. So concentration of your E. coli cells are 2 into 10 power 8 cells per ml. Calculate the multiplicity of infection. Let's see what is multiplicity of infection. When a group of cells, that is normal cells, are inoculated with virus particles, that is you are going to infect a normal cells with virus particles or any infectious agents, then your MOIS, that is multiplicity of infection, is number of virus particles present in your culture divided by number of target cells, that is your normal cells. So you are going to inoculate this phage into this E. coli culture. So multiplicity of infection is equal to number of virus particles divided by number of target cells. Number of virus particle can be calculated by multiplying the concentration of phages into volume of phage. Because here they have given concentration of phages as 4 into 10 power 9 ml power minus 1. That is, for 1 ml of phage has 4 into 10 power 9 number of phage cells. So we have to calculate for 0.1 ml. So for that we have to multiply 4 into 10 power 9 into 0.1 ml. So this ml will cancel it out. So 0.1 you can write as 10 power minus 1. Okay, so 9 minus 1 gives 8. So number of virus particle present in 0.1 ml is 4 into 10 power 8. Next, concentration of E. coli is given as 2 into 10 power 8 cells per ml. Then volume of E. coli culture taken is 0.5 ml. So we have to calculate number of E. coli cells present in 0.5 ml. Okay, so number of E. coli cells is equal to 2 into 10 power 8 into 0.5. So it means 1 ml has 2 into 10 power 8. So how much number of cells present in 0.5 ml? So 2 into 0.5 is equal to 1, 10 power 8. So number of E. coli cells present in 0.5 ml is 1 into 10 power 8. So multiplicity of infection is equal to number of virus particles, that is 4 into 10 power 8, divided by number of target cells, that is 1 into 10 power 8. So 10 power 8, 10 power 8 will cancel it out. So 4 divided by 1 is equal to 4. So MOI is equal to 4. Okay, next question. A microorganism isolated from a salt-rich lake, so salt concentration is approximately 2 molar, was found to possess diglycerol tetraethers with polyisoprenoid alcohol side chains as the major lipid component of its cell membrane. The isolated organism is. Okay, so here answer is an archaea. Okay, so the composition of archaea's cell membrane only has diglycerol tetraethers with polyisoprenoid alcohol side chains. Okay, it is a major lipid component of archaea's cell membrane. Okay, so the option D is correct. Next question is a list of pathogens in group 1. So group 1 has list of pathogens and a list of antimicrobial agents. So your group 2 has antimicrobial agents used to treat their infections. So these antimicrobial agents are used to treat this pathogens infection. Okay, you have to match the pathogens with their corresponding antimicrobial agents. Okay, so group 1 has pathogens and group 2 has antimicrobial agents. So you have to match which antimicrobial agents helps to treat these pathogenic infections. So the first option is influenza A virus. Okay, influenza A virus is a virus. So you have to find out the substance which has antiviral properties. Okay, so in these 5 options, amantadine, that is option 2, has antiviral property which is used to treat or prevent influenza A virus. Okay, and the second option is fungus. So option 3, fluconazole is an antifungal medicine which is used to treat infections caused by different kinds of fungus. And third option is plasmodium. Option 4, artemisinin is used to reduce the number of plasmid parasites in the blood of patients with malaria. So this medicine is mainly used to treat malaria and fever which is caused by plasmodium parasites. And the last option, mycobacterium. Okay, so isoniazid is an antibiotic which is used to treat mycobacterium tuberculosis infection. Match the human diseases in group 1 with the causative agents in group 2. So group 1 has human diseases and your group 2 has causative agents of these diseases. First option is amoebiasis. So amoebiasis is an infection in intestine. It causes diarrhea. Okay, then this disease is caused by protozoan parasite Endamoeba histolytica. Okay, so amoebiasis is caused by Endamoeba histolytica. This is a protozoan parasite. Here given all the causative agents are protozoan parasites. Then second option, African sleeping sickness. African sleeping sickness is caused by infection with the parasite Trypanosoma gambiense. Okay, then it is transmitted to humans by tsetse fly bites. Okay, this tsetse fly is only found in rural Africa. So African sleeping sickness is caused by Trypanosoma gambiense. Next one is Kala Azar. Kala Azar is caused by Leishmania denovani, then which is transmitted to humans by sandfly bites. It is Phyllocotomus argentifeis. The next one is Chagas disease. Chagas disease is caused by Trypanosoma cruzi, and this Chagas disease is transmitted to humans by insect vectors, and this insect is found only in Americas. Correct option is amoebiasis is caused by Endamoeba histolytica. Then African sleeping sickness is caused by Trypanosoma gambiense. Then Kala Azar is caused by Leishmania denovani. Then Chagas disease is caused by Trypanosoma cruzi. Okay, so 3, 4, 1, 2. Okay, so 3, 4, 1, 2. So option C is correct. The next question, which of the following processes can increase genetic diversity of bacteria in nature? So let's see what is genetic diversity. So genetic diversity causes individuals to have different characteristics. So within species you have different types of characteristics. For example, in apple species you have different varieties of apple, red apple, green apple, like that you have different varieties of apple. And in dog you have different breeds. So like that, in bacteria also you have different types of bacteria. So what causes the different characteristics within the species? So this is due to a genetic diversity mechanism. So in bacteria, naturally genetic diversity takes place by three processes. One is conjugation, second one is transformation, third one is transduction. Okay, so these three processes increases the genetic diversity of bacteria in nature. Okay, here the answer is PQR only. So option C is correct. Okay, let's see what is conjugation, transformation, transduction and transfection also. In conjugation, genetic material is transferred from one bacterium to another by cell-to-cell contact. In transformation, extracellular DNA is taken up by the recipient bacteria. And in transduction, virus, that is bacteriophage, transfers genetic material from one bacterium to another bacterium. In transfection process, foreign DNA molecule is delivered into eukaryotic cells. That is, extracellular DNA is delivered into eukaryotic cell in transfection process. So this process takes place in eukaryotes. These three processes takes place in bacteria. Next question is, which one of the following enzyme is encoded by human immunodeficiency virus, that is HIV genome? So in general, HIV genome consists of two single-stranded RNA molecules that are encoded within the core of the virus particle. Okay, so HIV genome consists of RNA in its head. So HIV genome uses reverse transcriptase to convert its RNA into viral DNA. And this process is known as reverse transcription. Okay, so in reverse transcription means the RNA is converted into DNA by using the enzyme reverse transcriptase. Next one is reference book for GATE Biotechnology Microbiology syllabus. So the first book is Microbiology by Pelzer, then Prescott's Principles of Microbiology. Okay, these two books are very useful for preparing microbiology syllabus for GATE Biotechnology. Next, let's see GATE Biotechnology Microbiology syllabus. Okay, so first topic is bacterial classification and diversity, microbial ecology. In microbial ecology, have to study microbes in marine, freshwater, then terrestrial ecosystem. Okay, then microbial interactions, structure and classification of viruses, methods in microbiology, microbial growth and nutrition. Then nitrogen fixation, microbial disease and host pathogen interactions, antibiotics and antimicrobial resistance. Thank you for watching my videos. If you like my video, please do share and subscribe.